All right, guys, so welcome back. Now we're doing a new series on homeostasis. So hopefully everyone's well. This is a really big, chunky topic, guys. So the video is probably going to last a while. You might not, if you're not feeling too keen at the moment, you might not want to go for it all at once. You can take it step by step. But everything's in there, guys. This is every single question that has ever come up on homeostasis in the BMAT till BMAT 2019. So without further ado, guys, let's make a move and start. So this is BMAT 2019, question 25. It says a student carried out tests to find out which molecules are present in a sample of urine from a human male. The student recorded the presence of large protein molecules in the urine and suggested that this indicated that the kidney was not functioning properly. And which of the following could have resulted in a high concentration of protein in the urine sample. So guys, pause the video, give yourself a minute, and make sure you're back. Where we're not only gonna go through the solution, but also the surrounding knowledge you need to have to answer this question. So guys, let's go through this. So before we even get into the question, let's just talk about how the kidney works really briefly, essentially to the level you need to know in the BMAT. So what you have is this part over here, which is this is basically the glomerulus. So you have the glomerular capillaries here. So you have the glomerular capillaries here. I'm just gonna write cap, and then here supplying the glomerular capillaries, you have the afferent arterioles, right? So remember, guys, you have artery, arterioles, capillaries, then venules, and then veins. But the kidney is weird in a way in that it doesn't follow that pattern. It's what we call a portal circulation. So you have an arteriole, a capillary, and then another arteriole. But this is the efferent arteriole now, right? Efferent arteriole. And the way I remember this is efferent is like the exit. So efferent is after the glomerular capillary. Right, so afferent is supplying the glomerular capillary, and the glomerular capillary feeds the afferent arterioles, and then this is surrounded by Bowman's capsule, right, which is where the filtrate essentially accumulates, and then we have a few structures after. We're going to go through these, and so I'm really bad at drawing, guys. Sorry, but hopefully this does the kidney justice. And remember, guys, this is not the kidney. This is actually called a nephron, which is the functional unit of the kidney, and you have around, I think, 1 million nephrons per kidney. So essentially, the idea is, guys, you force fluid out here, so that there's a net pressure forcing fluid out from the glomerular capillaries into the Bowman's capsule. And then from Bowman's capsule, you go into the next part, which is called the proximal convoluted tubule. So I didn't really do that justice there. So it says it in the name, proximal convoluted tubule. So proximal means like you're towards the start, right? And convoluted means it should be more like this rather than straight. So and then is a tubule because clearly it's just a tube, isn't it? So then the first kind of major part, the filtrate you would face then is the proximal convoluted tubule. And essentially what happens in the proximal convoluted tubule is you have selective reabsorption. So what essentially you do is you reabsorb all your glucose and your amino acid, and I think there's other ions like bicarbonate ions, but those details, as you don't have to know. Just remember the main ones. You're going to reabsorb all the glucose and all the amino acids, and the process by which that happens, guys, is secondary active transport. So you might have come across it actually in your A-level studies. Maybe you can just quickly go through that. So if you imagine this is like one of the cells here, right? And then we're kind of just zooming in on that. So on this side, you have, this is inside the tube, and this is outside the tube, if you like, in the tissue fluid, right? So what's going to happen then is what you're going to do here, you have a sodium pump, which you do 3 sodium out and 2 potassium in, and this obviously requires ATP, and then that means you have low sodium in the cell now, and essentially now you have in the case of glucose, you're going to have and also in the case of amino acids, you're going to have a symporter, so that's going to bring sodium in, obviously down its concentration gradient, because we have low sodium inside the cell now, and then along with it, it's going to bring in glucose or amino acids. It's essentially the same mechanism. Obviously, the channels are a bit different, but the mechanism is the same. So glucose and amino acids are actually going against their concentration gradient, right? But the fact that sodium is going down its concentration gradient, it can just drag glucose and amino acids along. So this is why it's called secondary active transport, because you had to do active transport here to then allow glucose and amino acids to go against its concentration gradient. That's why that's called secondary active transport. And then on this side, glucose and amino acids have their own transporters, which brings them out then into the tissue fluid, and then they're picked up by capillaries and then back into the blood. And those capillaries are actually just continuations of these guys. So these guys over here, the efferent arterioles just become what we call peritubular capillaries. So peritubular capillaries, and peri it just means near, and tubular means tube. So it's capillaries near tubes, and that's essentially what's reabsorbing all the stuff you reabsorb from the tube. Okay, so that's what's happening in the proximal convoluted tubule, and then you come down into this loop, which is called, has a special name, loop of Henle, right? And then in the loop of Henle, a critical part actually is this part over here, which is called the thick ascending limb, right? And essentially the purpose of the thick ascending limb is that it actively pumps out salt into the interstitium, or into the tissue fluid, if you like. So it pumps out salt over there, and essentially the purpose of that is to create a low water potential in the tissue fluid, or in the interstitium, if you like, right? So that's the role there. And then the part after this, let's just say around this part. Again, I'm not doing it justice in the drawing. It's called the distal convoluted tubule. So distal means towards the end, convoluted because it should be this shape, and tubule because it's a tube, right? And essentially the purpose of that part is it does a bit more essentially fine tuning. It further reabsorbs and secretes ions. That's just really fine tuning. And then the next critical part at the end is the collecting duct, so CD. So this part over here, this is critical. And essentially what happens here is that you can, because you from the thick ascending limb, you have created a low water potential in the interstitium. What you can essentially do is, via the action of ADH, which essentially makes pores, makes holes in this sort of membrane, you can essentially reabsorb water. And obviously, once you reabsorb that water, that will just go into the peritubular capillaries, which are going to be running around this whole tube, isn't it? Okay, so yeah, and maybe even, guys, if you really wanted to know the details, we can even just, we're not going to go super detailed, but the way ADH basically works is, if we draw one of those cells in the in the collecting duct, so let's just say one of the cells here, right? So then let's just say this is going to be inside the tube, and this is going to be outside the tube, and then obviously we have our peritubular capillary. This will deliver ADH, which is coming from the posterior pituitary. That's going to bring you ADH, right? And then ADH is going to bind to a receptor on the cell over here, right? And then that's gonna essentially lead to some molecular events. We're not going to go through that in great amounts of detail. And then essentially what happens is, it inserts channels over here. So channels are already present here, right, which allow water to go. Sorry, actually, wrong way around. Actually, so these channels over here on this side are already present. So those are ready to go. So already present, right? But what this signaling from ADH does is it causes these channels, and essentially normally these channels are present on the vesicles. So what the signaling does, it triggers this vesicle to essentially exocytose and fuse with the membrane, which is facing the inside of the tube, and then it inserts those channels. And then from that actually means you can bring water in here, and then water can then go through there, and then water can just go into the blood after that. And that's obviously going to bring the water potential of your blood back up. Okay, so I think, guys, that's really it. And then obviously out from here, that's basically the basis of the formation of your urine, right? So that's basically all the information, if not more, guys, that you need for this question. But let's go through this. It says damage to cell membranes between the blood vessels and Bowman's capsule. So can this cause a high protein urine? So also, guys, just to add, it the this is called ultrafiltration. So this thing over here is called ultrafiltration, where you're filtering stuff out from your blood into the Bowman's capsule. And this has 3 barriers. So the capillary, as you guys probably know, is lined by endothelium, and the glomerular capillaries are lined by a special endothelium, which is called it's a fenestrated capillary, essentially, which means that the endothelium has like little holes in it. And that obviously means that it kind of acts like a sieve. And then on top of that, you have like a layer here, which I'm kind of drawing there, and that's what we call a basement membrane. So essentially, all epithelial cells all epithelium cells are essentially lined by a basement membrane. So an epithelial cell is just a cell, which is essentially the difference between the inside and outside environment. So all these cells that are lining the nephron are epithelial cells, because if you follow the inside of the tube out, you lead to the outside world, and surrounding it, all of this is just tissue. So that's basically the definition of epithelial cells, isn't it? So that's going to be sitting on a basal membrane. That's really just as a scaffold to keep that epithelial cells in place. And there's something special about this basal membrane. The special thing, guys, is that it's negatively charged. And since it's negatively charged, that means it's going to repel negatively charged things from getting into the tube. For example, proteins, which are normally negatively charged in the blood. And then lastly, guys, you have the actual epithelium of the Bowman's capsule itself. And this has the structures called podocytes. You might want to Google these just to see how they look like. But these have like little slits in them, which further acts like it acts like a finer sort of sieve. So you have a 3-layer filtration system there. So if you damage those membranes, right, then obviously your sieve mechanism isn't going to work. So all these large proteins are just going to seep through into the tube, isn't it? And therefore, that could give you a high amount. It could give you the presence of large protein molecules in the urine when they normally shouldn't be there. So this is this can be right. And then it says a reduced rate of active transport and selective reabsorption from the nephron. So if so reduced rate and selective reabsorption, yes. So this guy's over here. Think about it. If you have reduced selective reabsorption, so remember, selective reabsorption is really talking about this guy, the PCT, right? So if you have less of this stuff going on, right, then that's obviously going to mean, guys, then that you're not reabsorbing. A, you're not reabsorbing sodium, and B, you're not reabsorbing glucose and amino acids. And what's that going to do to the inside of the tube is really going to bring down the water potential. And if that brings down the water potential, that's essentially going to attract water to essentially head into the tubes, isn't it? And therefore, what that would actually do is that would decrease. If water's coming in as well, you're going to have a large volume, and if you even have a large protein, it's going to have a large volume, and therefore that's going to act to dilute it, isn't it? So this one can't be the case. And also it says reduced rate of active transport, right? And if you have a reduced rate of active transport, that's really talking about this part over here, the thicker ascending limb, right? So if you have a reduced rate of active transport, then the salt is going to accumulate inside the tube, and the water is going to accumulate inside the tube. Large volume, that's going to act to dilute the protein concentration. So it can't be that one. And it says cells in the collecting duct do not have receptors to bind to ADH and so are unable to respond to this hormone. So if the cells in the collecting, that's what we're talking about here. So if the cells in the collecting duct can't bind ADH, right? So that means that you can't reabsorb. So you can't reabsorb the water, so the water stays inside the tube. And if that stays inside the tube, that would act to dilute again, isn't it, the protein concentration. So it says, which of the following can result in high? So obviously that would dilute it, so that would decrease the protein concentration. That has to be wrong. Therefore, guys, hopefully you can agree the answer would be B then, only one. Right? So hopefully, guys, that has made sense, and hopefully how the kidney works and the basis as to how the kidney works makes sense. And if you have any questions on this, guys, just post it down below in the comments. And guys, even if you want me to make a video focused on the systems of the body, because that's really what the homeostasis topic is about, I can make that. If you suggest it, I'm happy to make that, and if that's going to be of use to you. So hope to see you in the next video, guys. All right, guys, so welcome back. So previously we were looking at BMAT 2019 question 25, where we were going probably even in a bit too much detail, guys, about how the kidney works. But hopefully, guys, you follow that up, and hopefully that makes sense. Remember, guys, if this is at all confusing, don't be shy. Just post questions down below, and we're all happy to help each other. So next, guys, we're going to go on to BMAT 2018, and this is question 1. So let's read it together. It says, people with cystic fibrosis may be unable to digest their food fully because of their thick, sticky mucus. This prevents the secretion of pancreatic juices. Doctors sometimes prescribe enzyme supplement medication to improve digestion. It says, which of the following enzymes should be included in the medication? So guys, pause the video, give yourself a minute, make sure you're back. We're going to go over not only the solution, but also the information around this topic that you have to know about. Okay, guys, so let's welcome back. So firstly, cystic fibrosis, just giving you a bit of general information. Essentially, what cystic fibrosis is you have this special channel, which is called the CFTR channel. I think it stands for cystic fibrosis transmembrane regulator. So that channel is messed up. So if you like, if you imagine you have a tube, let's say this is the tube that essentially is coming from the pancreas and is going to duct into the small intestine, and where the pancreas can really secrete its enzymes and all its solutions into the small intestine. So obviously this is an epithelial cell over here because it's the if you follow this tube in, so obviously if you follow this pancreatic tube, it's going to lead on to the small intestine. If you follow that out, it's going to lead to the outside world. And also this is just bounded by tissue. So that's basically the definition of an epithelial cell, isn't it? So that's going to be an epithelial cell over there. And normally this channel there, the CFTR channel, what it does, it secretes choriodines into the duct, right? And then what's that going to do, guys? That's going to lower your water potential. And therefore, what that should do is it should push water in to the tube into the tube as well. But then if this is messed up, guys, now what that's going to mean if that's messed up is that you're not going to secrete water, and then therefore if you imagine your secretions are going to be really thick, they're not going to be watery and they're not going to easily run, and therefore when it's thick, it's really going to block the tube. And when it blocks the tube, so if you imagine this is like the first part of the small intestine, right? So if it blocks the tube, then the stuff that the pancreas is secreting can't reach the small intestine. And if it can't reach the small intestine, you're going to have problems digesting stuff. Because remember, the pancreas puts into the small intestine enzymes literally required to digest all the macromolecules, so carbohydrates, lipids, and proteins. And really what this is testing, and this is a topic based on digestion. So it's really testing is if you know where the enzymes to digest the macromolecules come from. So the way I normally remember it, guys, is just remember that each of the macromolecules are digested in two places. So carbs, right? So where carbs are digested is the mouth plus the small intestine. Small intestine, but the enzymes are coming from the pancreas, right? And then you have protein, right? So protein is mainly in the stomach, right? So you have the stomach and the small intestine, but those enzymes again are coming from the pancreas. And then you have lipids, right, or fats. That's basically mainly going to be in the you actually have a bit, but it's really bad in the stomach. I'll explain why that is, but mainly in the small intestine. Essentially, the reason, so if you imagine the stomach is something like, let's just say this is the stomach, this is the tank in your stomach, right? And then obviously into your stomach you have acid, you have enzymes, and all these solutions. That's going to be in sort of the solution phase. So enzymes are also going to be in here, and the stomach. There are actually there are gastric lipases or stomach lipases present, which can digest lipids, but they're in this phase. But then obviously if you do the oil and water experiment, what normally happens, the water sinks to the bottom, and then you have like a layer of fat or oil on the top, isn't it? Now, can you see the problem there, guys? Because the enzymes in the solution phase, it can't really nibble on the lipids as much because they're not really mixing, and the mixing only really happens in the small intestine with the help of bowel, isn't it? Which is being released from the gallbladder into the small intestine, right? So, but it can happen a bit. I mean, I hope you can appreciate, guys. It can happen a bit, but not fully, right? Um, so yeah, if this mechanism is messed up over here, you're not going to get you're not going to get so the enzymes which break down carbs and further break down carbs is going to be, you're not going to get pancreatic amylases, you're not going to get proteases, right, or trypsins, stuff like that from your pancreas, and you're also not going to get lipases from your pancreas. So in this medication, therefore, should be all of these stuff, isn't it? Because if your pancreas is not working, none of these are going to produce, and therefore you need to supplement it with all of them. So the answer there for guys, hopefully you appreciate the answer, would be F. So hopefully, guys, that has made sense and hasn't caused too much problem. And I look forward, guys, to seeing you in the next clip. All right, guys, so welcome back. So previously, we're looking at this question about cystic fibrosis. We went in a bit of detail about what causes these symptoms of cystic fibrosis and linked this to the topic of digestion. Really, guys, that detail about what we just given about how the digestive system works is really what you need to be knowing, and nothing much more detailed than that. But be sure, guys, to read the specification because it changes from year to year. But next, as we move on, BMAT 2018, question 17, and says the diagram shows a simple piece of apparatus that can be used to demonstrate some of the events involved in human ventilation. And then you have the apparatus that says which event occurs during ventilation will be demonstrated by pulling the rubber diaphragm downwards as shown by the arrow. So guys, pause the video, give yourself a minute, and be sure to come back. We'll be going through the solution. Okay, guys, so let's just look at this apparatus and link this in essentially to so this is clearly a long question, isn't it? Because it's talking about ventilation. Let's link this to what would be the equivalence of each part in the actual human body. So the rubber diaphragm, they basically give you a clue already in the name. This is basically going to represent the diaphragm, right? And then what else do we have? So then on top of that, guys, we also have this, which is going to obviously represent the lungs and going to be the associated tubule. So this is going to be the bronchus, I'm just going to write bronchus plus. So so you have the bronchus, the bronchioles, and they further divide. And then obviously the big one feeding in from the mouth is going to be the trachea. That's going to be the trachea. And then the wall, guys, this is going to be essentially your chest wall, isn't it? So the membranes which are associated with the lungs, and then you have the rib cage and the associated intercostal muscles, isn't it? And this is actually a good depiction. So it says when you put the rubber diaphragm down. So when you put the rubber diaphragm down, what's going to happen to the volume inside this cage, or in the human body? Obviously, this will be called the thoracic cavity, right? So when you put that down, what's going to happen is that uh, you're going to increase the volume, right? And then there's a law in physics which says volume and pressure are inversely related. So if you increase the volume, you're going to decrease the pressure. And this is all relative to the atmospheric pressure. You're going to have a lower pressure than the atmosphere. And essentially what happens then is therefore the lung expands. So if you imagine the low pressure is like a suction, it's really going to suck this, and then the lung is basically going to expand. And therefore the lungs going to expand to fill up that space. So therefore, what's going to happen then? The lungs would therefore again have a high volume, low pressure. Again, this is all relative to atmospheric, and that low pressure is going to draw air in from the outside world down a pressure gradient, isn't it? So from a high pressure in the atmosphere to a low pressure inside the lung. And therefore that's going to allow you to breathe in, isn't it? And this is really what happens, guys, when you're breathing at rest. So when you're breathing at rest, like probably now, hopefully this video isn't causing you guys to panic, but if you're breathing now, your chest, if you put your hand on your chest, it shouldn't really be moving much. It's mainly just your diaphragm which is doing all the work when you're breathing at rest. So it says which events occurring during ventilation will be demonstrated putting the rubber diaphragm downwards as shown by the diaphragm, as shown by the arrow, sorry. So when the diaphragm goes down, that's essentially represents diaphragm contracting. So when any muscle contracts, and obviously the diaphragm is a muscle, isn't it? So when any muscle contracts, essentially it shortens. So when it's relaxed, the diaphragm is like that, and then when the diaphragm is contracted, it's more like that. And can you see this has to be the contracted version, guys? Because if you measure the distance from here to here, or from here to here in the contracted version, that would be shorter. And that's obviously makes sense because I said when you contract it, the muscle gets shorter, right? So that means the diaphragm is contracted, so it can't be that one. And then what's happening to the volume of the thoracic cavity? We already said that, isn't it? So the volume is increasing. So we've got that. And then what's happening to the pressure? So the inverse related, so the pressure should be decreasing. So we got that. So then the answer, guys, should be 2, 3, and 6. And that maps on nicely to F. So hopefully, and that's really it, guys. That's really all you have to know about how the lungs work. I think you also need to know a bit about the intercostal muscles. So just remember, so external is inspiration. So let's just write this over here. So the external intercostals is for inspiration, and the internal intercostal muscles is for expiration. It's kind of like the opposite. So you think external, E goes with E and I goes with I, but it's actually the opposite way around. That's how I remember it. And really, guys, that's really it in terms of ventilation. So hopefully, guys, that has made sense, and I look forward to seeing you in the next clip. All right, guys, so previously we are looking at this question to do with a breathing mechanism and how it relates to this apparatus. Hopefully, guys, that has made sense. But now we shall move on to the next question, which is BMAT 2017, question 1. It says the diagram shows a section through part of a healthy human. It says which row on the table shows the correct secretions from P, Q, and R. So, guys, pause the video, give yourself a minute, and be sure to come back. We'll be going through the solution. Okay, guys, so let's just go through this. So let's just actually look at the anatomy and look at what things represent what. Right, so hopefully, guys, you can probably see this line over here. So what this line is representing over here is the diaphragm. So that's basically the diaphragm is the separation between the abdomen, the tummy area, and the thorax, which is the chest area. And then can you see, guys, obviously the esophagus, this is the esophagus, which is poking through the diaphragm as it comes, it goes from your mouth, and then it comes down your thorax, and then eventually through the diaphragm into the abdominal cavity. And then this feeds into this structure over here, which is going to obviously be your stomach. Right, so that's your stomach, and this is right there. That's your esophagus. I'm going to write also, and then from your stomach, this leads on essentially to this part, which is going to be your small intestine, and then that loops around, and then around the perimeter, that becomes the large intestine, and then that's the large intestine, and then after that becomes your rectum, and then eventually your anus, and then you poop your stuff out, isn't it? And then this structure but on the diagrams left but our right, so on our left, if you're looking at it on the screen, but the patient's right, this would be the liver, isn't it? So this is going to be the liver, and associated with the liver over here is going to be the gallbladder. So this is going to be the gallbladder. So the gallbladder, remember the function of the gallbladder is to essentially hold on to bile, excess bile that the liver secretes into the small intestine, and it also acts to concentrate the bile by sucking out water and making it more concentrated, right? And that's why I think if you have too much of these secretions happening, bile, you can get stones in your gallbladder, and those are really painful. And then it says, which one on the table shows the correct sequence from P, Q, and R? So what does P secrete? So we just said that, isn't it? That's bile. So we know this is bile. So we know it's that. Does it secrete a high hydrogen ion concentration? Actually, no. That's more of a thing of the stomach, isn't it? So it can't be B. This one doesn't have bile, so it can't be that. This one doesn't have bile. Yeah, and we basically already got an answer. But let's just go over the rest, guys. So then Q is the stomach. So remember, the stomach secretes proteases. It also secretes some of the main things it secretes is proteases, which bring up, that's the main thing. A little bit of lipases, but that's not really. I don't even think they mentioned that at GCSE level, but that's something to be aware of. High hydrogen ion concentration because it's really acidic, isn't it? And also, it releases something called intrinsic factor, which is required for B12 absorption. But even that you don't have to know. But just remember proteases and hydrogen ions. So this is good. And then R, what's R? So R, oh, we forgot about R. Remember, R is the pancreas, right? This is the pancreas, and this this tubular-like structure over here is going to be called the P duct or pancreatic duct, right? And can you see, essentially the stuff from the gallbladder? So the stuff from the gallbladder and the stuff of the pancreas essentially enter the small intestine at approximately basically the same location at the beginning parts of the small intestine, right? And obviously, yeah, so what the pancreas releases is all of the enzymes which are going to digest all the macromolecules of proteins, lipases, amylases, and they also release, so that's their sort of exocrine function, but they also have an endocrine function, which is to do with hormones. And we're going to go over this probably in a future question when it comes up, insulin and glucagon. So insulin from beta cells of the islets of Langerhans, and glucagon from alpha cells of the islets of Langerhans. So this is the last option, and yeah, that's really hopefully, guys. Yeah, so the answer therefore is good, and is A, and hopefully you can understand that. So I hope you guys has made sense, and I look forward to seeing you in the next question. All right, guys. So previously we were looking at this question, which was to do with how the digestive system essentially works. Hopefully that has made sense, guys, and that was really a good revision. Again, I hope of how the digestive system works. But now, guys, we move on to the next question, BMAT 2017, question 17. Says the diagram shows an external view of the human heart, and then we have an artery present on the surface of the heart. It says, which of the following statements about the artery shown on the diagram is or are correct? So guys, give yourself a minute. Make sure you come back. We're going to be going over the solution. So guys, hopefully you had enough time to go over this question. So firstly, we just need to identify what that artery is in it. So the artery on the surface of the heart, likelihood is that it's probably going to be supplying the heart, isn't it? And that's the job of the coronary artery. That's it. So the coronary artery, and coronary, I think the word, it has something to do with the heart. So it's the artery of the heart. And essentially, the way this essentially is forming is that, so obviously, guys, hopefully about the arterial supply and the venous supply to and from the heart, right? So you have the vena cava supplying blood in, then pulmonary artery, blood out to the lungs, oxygenate, pulmonary veins, blood back to the heart, and then you have the aorta, which pumps it out to the rest of the body, isn't it? So you basically have so imagine this is the aorta, right? And then immediately coming from the aorta, you have this is basically the root of the coronary vessels. So basically what normally happens is really cool. So when the left ventricle contracts, it's basically going to force blood to go in this direction, isn't it? But then when the heart relaxes, it kind of creates a suction to draw blood back. And when the blood is drawn back, it can kind of enter these areas. It can enter these areas through here, because obviously when that happens, when the heart's in diastole, the aortic valve, which is the valve between the left ventricle and the aorta, that's going to snap closed. So when that snap closes, the blood therefore shifts sideways, and this goes into the coronary arteries. So the coronary arteries, right? So then let's go through this. Is the site of the diffusion of glucose and oxygen into the muscle cells of the heart. Okay, this is a really trick question. So you would think that because it's supplying the heart, but it's supplying the heart. But the place where actual exchange happens is the capillaries, not the arteries. Remember, so you have the arteries which are just supplying the capillary beds, and the obviously from the arteries you have arterioles. And really, what arteriole is, guys, is just a sort of control point. So if you imagine like you have a big artery, and then this is going to supply many arterioles. Let's say it's supplying 3 arterioles, right? And basically, what arterioles are just little control points, essentially. So they have a ring of muscle around them, smooth muscle, right? And essentially, they can. So let's just say, for example, this is organ 1, organ 2, and organ 3. And let's just say, for example, this is the gut. This is the, let's just say, this is going to the toes, and this is basically going to the muscle, right? If you're exercising, guys, you probably don't want to be sending much blood to, for example, the skin of your toes. You don't really care about that. Neither do you want to be sending much blood to your gut. Mainly, it wants to be to your muscles, isn't it? So what normally happens is this smooth muscle and that smooth muscle contract. Remember, when they contract, it's a circle. When it contracts, it's going to get shorter. So essentially, the circumference is going to get smaller. So they're going to really decrease the lumen size. So you're kind of blocking this port, right? You've blocked this port, and then now essentially all the blood that's coming in is just going to go. So really, arterioles is just little pressure control points. Arteries supply the arterioles. Capillaries is where you do all the diffusion and stuff, and then you have venules and then veins eventually go back to the vena cava. Right, so they are not the site of diffusion, isn't it? That's the job of capillaries. And it says it transports blood at high pressure. Yeah, because remember, essentially, arteries are essentially they're closest to the heart. If you're closer to the heart, right, so essentially the vessels which come immediately after the heart are going to have high pressure, and then as you go along the tree, you basically get low pressure until you get to the veins, where the pressure is really quite low. So it transports blood at high pressure, and that's true because arteries are close. So arteries high pressure, and then veins essentially low pressure, and then it contains muscle cells, and that's also true. So this basically is talking about. This is just talking about if you know the structure of. Can you distinguish between the structure of arteries, capillaries, and veins? So arteries and arterioles. So let's start with arteries. So arteries have a structure where it's basically you have an inner layer of endothelium, right? So endothelium is kind of like epithelium but for blood vessels, and then surrounding this, guys, you have a layer of smooth muscle, and also in arteries you have a bit of elastic fibers because they really have to maintain a constant pressure. You probably heard of that in your A-level studies. So they're elastics to make sure the pressure doesn't deviate much. So they have so you have a layer of muscle and elastic fibers, and then around that you have a layer of connective tissue, right? And essentially what the connective tissue does, it says in the name, it just glues the vessel into the tissue so it remains in place. So that's basically the arteries. The arteriole is basically the same thing, but since the artery is already doing the pressure regulation, it doesn't have to do much pressure regulation. In terms of, it's not going to experience huge deviations because the artery is already doing that job. Therefore, in the arterioles, you mainly just have muscle in the second layer. And then the capillaries, what the capillaries are, is just literally the inner layer. So a capillary would look somewhat like that, right? And then essentially the vein is just a low-grade version of an artery. So it basically has all the layers of artery, but to a much lower extent, right? So it has all of them have endothelium. It has muscle, but it has really little muscle, and it has connective tissue, again, really little connective tissue. And the main thing is that the veins are much bigger. So if I just draw a circle, a vein, the size of the lumen of the vein will be that much. And since you have a high volume, remember high volume and pressure inverse rates, if you have a high volume, you're going to have a much lower pressure there, right? So therefore, guys, hopefully you can understand then 2 and 3 are the correct answers. The correct answer would be G, right? So hopefully, guys, that has made sense. And if you have any questions, post down below, and I hope to see you in the next question. All right, guys, so welcome back. So previously, we were looking at this question to do with the coronary arteries. Hopefully that has made sense, guys. And any problems at all, if ever, just make sure you get into contact with me. Now, guys, we're going to move on to 2016 question 1. It says, the diagram shows a kidney and its associated vessels from a healthy human. And it says, what role correctly identifies the vessels along with the concentration of urea they contain? So, guys, pause the video. Make sure you give this question a try. Give yourself a minute and make sure you're back after that minute where we can go through the solution. Okay, guys, welcome back. So let's go through this question. So this actually ties in really nice with the previous question because there's a difference between. Hopefully, you can appreciate, guys, that these two things are blood vessels, but the question is which blood vessels are they? But can you see with this one over here, the lumen is much bigger. So that tells me then this is a vein, And since this is a vein to do with the kidney, it's going to be called a renal vein, right? And therefore this one must be, that's good, that one must be a renal artery, right? Or probably actually not an artery yet. This is actually the main vein which is supplying essentially the main vein which is draining from all the organs. So this is actually the vena cava, and that's going to be the main artery which is supplying all the organs, which is going to be the aorta, right? And then once you actually, yeah, that's good. So once you get into here, this is now where you're in your renal artery, right? Because essentially what the aorta is just one long tube, and then at regular distances it gives off branches to supply the various organs, right? And then this is the vena cava, and in particular it's the inferior vena cava, because essentially this is draining blood from everything which is below the diaphragm. And the proper name for the aorta here would be the abdominal aorta, because the kidney is in the tummy area, and therefore yeah, hence why it's called the abdominal aorta. And then so you have renal artery there, and therefore this one over here would be the renal vein. So hopefully you can appreciate that. And then you obviously have the kidneys, and then what comes out of the kidneys? This thing, guys. So what comes out of the kidneys is, guys, is the ureter. So this is also really important in terms of the BMAT. Always comes up. So from the kidneys is the ureter, and then the ureters drain into this structure here, which is the bladder, which holds about I think just under half a liter of fluid, and then from there comes out from there is one tube, which is called the urethra. And obviously, females have a shorter urethra compared to males, and essentially the urethra is essentially where you pee out from. Right, so I think that's all the structures. And then urea, what's the significance of urea? So basically, urea is the main waste product we produce. So protein breakdown, essentially. So proteins obviously have amino acids, and amino acids, the main component of the amino acids, you have the ammonia group, isn't it? And essentially, the ammonia groups are converted into urea, right? And then urea is what you release out from your body. That's how you excrete the breakdown products of proteins, if you like. And obviously, we break down a lot of protein every time. So where are we going to have the lowest concentration of urea? So hopefully, guys, you can appreciate that the lowest concentration of urea would be number 3, actually. So the reason why I think it would be number 3 is this, because that's fresh. That's so the renal vein is blood which is freshly coming out of the kidney, isn't it? And it's just after you've taken out all the urea. So literally, but at really close to this point over here, you're basically going to have no urea in the blood. But then obviously, as soon as you get into one, remember the vena cava is draining blood from various organs. So let's just say, for example, the liver might duct over here, and then by this point, you might have added. Yes, you've cleared it up at this point, so just to say you have 0 here, you have 0 here, but then by the time the liver duct in, you might suddenly, for example, get 100, because the liver processes, liver essentially is the place where you make the urea, and that might just throw a lot of urea into the blood. So that's why I think it's the renal vein. And then where you're going to have the highest concentration, so you're going to have the highest concentration in essentially your urine, isn't it? So that's why you're getting rid of all of it. And the most distal, so the last, so the things which are after the kidney is where you're going to find the highest concentration of urea. And in this case, that's going to be number 5, which is the ureter. So that's going to be this one, isn't it? So number 5, and then yeah, 3. Let's just make sure that's the renal vein. Yeah, that's it. That is the renal vein. So the answer, therefore, guys, it would be D. So hopefully, guys, you can really appreciate why D is the answer. And I think that was a good question in terms of revising the. So this is really about the macro structure of the kidney, and we looked at the microstructure, I think, in an earlier video, and we were talking about how the nephrons work. So hopefully, guys, that has made sense, and I look forward to seeing you in the next clip. Okay, guys. So previously, we were going through BMAT 2016 question 1 to do with the O structure of the kidney and a bit about urea. It's really important, guys, these concepts. I think the kidneys especially is quite a common topic they like to ask in the BMAT. So hopefully, guys, you understood that. And if there's anything you don't understand, just be free to send messages down below. But now, guys, we move on to BMAT 2015 question 1. It says, which one of the following is not part of a reflex to the stimulus of placing your hand on a hot object? So, guys, pause the video, give yourself a minute, and be sure to come back. We'll be going through the answer. Okay, guys, so let's try to at least draw a picture of the hand. So this is my picture of a hand, something like that. And essentially, guys, in your hand, you have a load of sensory neurons, and they essentially do what they say on the tin. They essentially sense stuff. Where there'll be specific ones for temperature, specific ones for pressure, specific ones for pains, etc., etc. So you have these sensory neurons like this, and these are generally quite long, right? So these are generally quite long. And then I'm also going to draw in here a spinal cord or a section of a spinal cord. The reason why I'm drawing that would make sense in a little while. So let's just say that's a picture of a spinal cord, right? So that's the picture, or that's my attempted picture of a spinal cord. And essentially, the sensory neuron, right, has a cell body. If you guys don't know what cell body is, you're definitely going to cover it in the second year of A levels, or some of you might have even covered that at GCSE. But what cell body is where all the machinery of a general cell is, like mitochondria, ribosomes, nucleus, and stuff like that. For a neuron, it's all in the cell body. But then from the cell body, you have one branch which is going to the skin of the hand, which is the sensory branch, if you like, and then you have one branch which is actually going to the spinal cord, and this synapses with a neuron which is actually within the spinal cord, and this neuron is called is a small neuron. It's called a relay neuron. And essentially, a relay neuron says, as it says on the tin, it's going to relay information from the sensory neuron onto the next structure, which is this guy over here. And this neuron over here is present on the ventral surface of the spinal cord. So ventral just means towards the front. So the front of your spinal cord, right? So if you touch your tummy, so that's your front of your body, and the back, the special name for back would be called dorsal. So the back is on your back basically, and then your ventral is on your tummy side. So neurons from the tummy side of the spinal cord are going to be called motor neurons, and these neurons are going to send information down then. So this is going to be MN, motor neuron, and then in the case of when you touch a hot object, this is going to send information, this information down to your biceps, telling it to contract. And when your bicep contracts, you quickly retract your hand away from the hot stimulus, and basically you're saving your tissues. This is the purpose of these reflexes, in it, to protect yourself. So guys, so hopefully. So hopefully you can see that. So you have sensory neuron, which is super long from your hand, and it's going to essentially go into your spinal cord. So that's a bit of a distance, isn't it? And then the relay neuron, which is really small, which is really just within the spinal cord. And then you have a motor neuron, which is so in this case over here, obviously it's shorter than the sensory neuron, isn't it? Because it just has to go from the spinal cord then back to your biceps rather than all the way to your hands, right? So that's much shorter distance. So the reason I'm saying the orders, guys, is this is just a hint, heads up for a question which is coming up, which a lot of students, my previous students, have got wrong. But hopefully you guys won't make the same mistake. But let's go through this. And there's obviously synapses, isn't there, at every point. Remember, synapse, guys, is just a point of chemical communication between neurons, where you change the information from electrical, which is how it's passing through neurons, to chemical, and then back to electrical in the next neuron. So then it says sensory neuron transmit electrical impulses to the central nervous system. That's true, isn't it? So the mode of information, the way they carry information is through electrical impulses, and they are going to the spinal cord, which is part of the central nervous system, isn't it? Central nervous system is the brain and the spinal cord. So this one's good. Oh, sorry, it says which one is not part of the reflex. Okay, so that one's going to be wrong, isn't it? And then it says muscle cells contract. Yeah, that's true, but they are part of the reflex, so this one can't be it. Motor neurons transmit electrical impulses to muscle cells. Yes, that's true. So it can't be that one. Relay neurons pass electrical impulses from sensory to motor neurons. Yes, that's clearly what we've just shown there, isn't it? So it can't be this one. Process of elimination, therefore, is going to be E. But let's just prove that this is wrong. Brain transmits electrical impulses to relay neurons. That's wrong, isn't it, here? Because remember, in a reflex, you want it to be really fast. You don't want to be sending information from your hand to your brain and then back down. That's really going to waste time, and you're really going to damage some tissues if you actually do that kind of thing, isn't it? So that's why the brain is not involved in a reflex. So hopefully, guys, that has made sense. And really, guys, be careful when it says which of the following is not part of the reflex. I nearly made the mistake, but then I realized that then it's really bolding this thing out. So hopefully, guys, that has made sense, and I look forward to seeing you in the next clip. All right, guys, so welcome back. So previously we were looking at BMAT 2015 question 1. So hopefully, guys, we have revisited effectively the way reflexes work, and just a bit about a bit of a touch about how the nervous system works as well. And hopefully you managed to follow up by Eva's correct answer. But now, guys, we move on to BMAT 2015 question 13. This says a human consumes the same amount and type of food and drink on 2 consecutive days. He also does the same activities on both days. However, on one of the days it is hot and the other it's cold. Which row on the table correctly shows the mass of substances found in the urine of this human on the hot day compared to it found on the cold day? Okay, so guys, pause the video, give yourself a minute, be sure to come back. We'll be going through the solution. So guys, let's go through. It says mass of substance found in urine on a hot day compared to the on a cold day, right? So what would what's going to be different between a hot day and a cold day, guys? So the main difference is essentially on a hot day, physiologically, what's going to happen to you is that you're going to sweat more, isn't it? So you're losing more water, right? And therefore, your body is going to act to conserve water, isn't it? And if your body is acting to conserve water, remember the key hormone you're thinking about is ADH, right? And remember, when you have ADH present, right, what that's going to do is going to create, essentially, it's going to create holes in your collecting duct. And essentially, when it creates, so let's just say this is your collecting duct, it's going to create holes in your collecting duct. And let's just say, for example, obviously this is just bounded by tissue, right? So it's going to create holes in your collecting duct. And then what that's going to allow then is for water to seep out of the tube. And remember, around the tubes, you have peritubular capillaries, and then the water is going to be reabsorbed by the peritubular capillaries, and it's going to go back into your blood. And essentially, you're going to maintain a constant osmotic potential or water potential in the blood, isn't it? So if the water is seeping out of the tube, therefore, what's left in the tube is going to be less water, isn't it? So therefore, you're going to have less water in your urine on the hot days. So that's going to be less. So we've already knocked out quite a few options. And then what's going to happen to the urea? So in terms of the urea, guys, is talking about mass here. So so this is really a good question. So if it talked about concentration, right, the concentration of urea would be more, isn't it? Because you have essentially, since this is the key thing, is he's having the same amount and type of food, right? So we can therefore say that he's having the same amount of proteins and stuff like that, and same amount. He's going to essentially produce the same amount of nitrogenous waste. Remember, nitrogenous waste is secreted out from the kidney and the rest of the body through the form of urea. So he's going to be making the same amount of urea, but then if it was concentration, the urea will be more concentrated because there's less water inside the tube with the same amount of urea. Remember, concentration equals moles divided by volume. So if the moles is staying the same because the mass is staying the same, remember moles is just mass over mR. So the mR for urea is not going to change, the mass isn't going to change, therefore the moles is going to change, and that's going to be constant. But obviously, since the volume of urine is going to be lower, the concentration of urea will be higher. But here it's just asking for the mass. So the mass is going to basically be the same because of his eating and drinking the same. So therefore, guys, hopefully you can appreciate that B would be the correct answer here. So guys, I really hope that has made sense, and I look forward to seeing you in the next question.